Hello, dear students. Today we will continue with the next topic of circulatory system. We had done the RBCs in the previous video. So today we will be focusing on white blood cells or the leukocytes. So let's start. The blood cells are formed in the bone marrow by a special category of cells which are called as stem cells. So some of these stem cells lead to formation of red blood cells, platelets, and the granulocytes, one category of WBCs having a granular cytoplasm. Another category of these stem cells enters the lymphatic tissue for maturation, and then they are called as the agranulocytes, in which we have the lymphocytes and the monocytes. There is also this natural killer cell which lies in a dormant state in the blood plasma, and it only becomes active when it encounters any danger or any threat to the immunity or the health of the well-being of an individual. The B lymphocytes are those lymphocytes which mature in the bone marrow only, whereas the T lymphocytes are those lymphocytes which mature in the lymphatic tissue, especially of thymus. So let's study these properties of WBCs in detail first. So first thing, the bone marrow, red bone marrow, the lymph nodes, liver and spleen, they are the areas where WBCs are produced. The liver is the largest organ in the body. The spleen is a structure behind the stomach. The lymphatic nodes are those areas where WBCs are produced. They are present, distributed on your entire body in various regions. They are concentrated, but they are again the area where WBCs are produced. And then we have the bone marrow. Amongst all bone marrow, some part of the bone marrow is yellow and some is red. The WBCs are made in the red bone marrow. So the yellow bone marrow is basically made up of fatty tissue. As we age, the amount of yellow bone marrow increases and the amount of red bone marrow decreases. The bone marrow, which is red, only remains on the corners of long bones. So this particular site, the WBCs are synthesized. Another thing about WBCs, they are about 4,000 to 8,000 per cubic mm in blood. The volume is much less. The concentration is much less as compared to the concentration of RBCs. RBCs are present at, there are roughly 6 to 7 million RBCs per cubic mm of blood. RBCs are required because they transport oxygen. So their requirement is much higher as compared to WBCs, where their basic requirement is for immunity, for taking care of body's health against diseases. Whereas for all vital activities, oxygen is needed. That is why concentration of RBCs is much higher. Their average lifespan is about 2 weeks. The average lifespan of RBCs is 120 days, but for WBCs, it is only 2 weeks. And amongst all WBCs, one category of WBCs, which we call as neutrophils, their lifespan is about a few hours only. They are very active in immune responses. They fight germs better, so they are more active and their lifespan is shorter. They are destroyed in spleen, in liver, and in bone marrow. Another thing about the WBCs is their abnormal increase is called leukemia. Leukemia is a disease which is fatal. It's also called as blood cancer. Normally, there are more RBCs and a fewer WBCs in a slide, if you happen to examine a slide under a microscope. But when the number of WBCs become much higher, it will increase the number of WBCs at the cost of RBCs. So the oxygen carrying capacity of blood reduces. It will affect the functioning of vital organs, and thereby overall functioning of the body goes down. This disease generally results in death. There is a cure of blood transfusion, but that again may not be a certainty for everybody. So one disease which is fatal is called leukemia, which is blood cancer. Then if in case there is an abnormal decrease in the number of WBCs, it's called as leukopenia. And they display two important processes. Number one is phagocytosis. That means they eat the germs when they encounter any germ or any infection. They eat the germs. And the second one is diapedesis. WBCs are amoeboid in shape. They have an irregular shape. So they squeeze out of the blood capillaries, and in doing so, they move out, squeeze, distort their shape, and that movement is called as diapedesis. Now, I told you with advancing age, the red bone marrow decreases and it is replaced by yellow bone marrow. Now, when we are younger, almost all bones have red bone marrow. But as you start aging, the amount of red bone marrow decreases and it is taken over by yellow bone marrow. So much so that the red bone marrow only remains in a few bones. And it is these bones where the WBCs and RBCs are synthesized. So if you happen to see here, this is the outer surface of a bone. This portion is called as the red bone marrow, and this here is called as the yellow bone marrow. So the yellow bone marrow remains in the middle and the red bone marrow remains on the sides. But as we age, the red bone marrow slowly and slowly finishes off and it is replaced with yellow bone marrow. Yellow bone marrow is rich in fatty tissue. So it obviously is not fit for synthesis of RBCs or of WBCs or of platelets. So here is how it is synthesized. So in case of children, we already studied that the RBCs are made in bones of all bones. But as you age, the RBCs are made in long bones. And even WBCs are made in long bones. So this is only because they have still got red bone marrow in them. Now we did the term diapedesis. Diapedesis is displayed by WBCs. WBCs, when they're moving in the flow with the blood, they encounter any germ substances. So these WBCs come to the inner lining of the blood capillary. They stick onto the inner lining, and wherever they encounter an opening, they squeeze and distort themselves to come out of that opening toward the place where germs are accumulated. This squeezing out is called as diapedesis. This is only possible because of the amoeboid shape of the WBCs. And this is an important process displayed by all WBCs. Phagocytosis, on the other hand, is a process of eating germ substances. In phagocytosis, the leukocyte, it absorbs the bacteria. The moment after diapedesis, the leukocytes leave a cell, they absorb the bacteria. After they've absorbed the bacteria, they digest the bacteria. After digesting the bacteria, they expand because they don't digest just one bacteria, but a large number of bacteria. When they expand, their cell volume increases more than the normal size, which will lead to the bursting or the lysing of the bacterial cell. When the bacterial cell will lyse or burst, it will release chemicals called as cytokines outside. These cytokines attract more WBCs, more leukocytes to the site where some kind of bacterial accumulation was there. This will ensure that these WBCs ultimately destroy any germ substance which still remains in this area. Now, when the WBCs ingest the bacteria are ingested, it increases in its volume and it releases these cytokines. Because of release of these cytokines, more WBCs will be attracted. When more WBCs are attracted, it causes a process. It leads to a triggering of a reaction, which is called as inflammation. When any injury happens, the harmful substances, they enter your body, the WBCs will eat them by the process of phagocytosis. They will be releasing cytokines, which will ultimately attract more WBCs to that area. When the number of cells in that area will increase, it will lead to swelling, redness, and obviously because of increased muscular activity, there will be warmth. You will have a sensation of pain because injury took place. So pain receptors in the skin, they will be triggered. So this entire thing is called as inflammation. Inflammation is body's response to a particular infection, and it will ultimately lead to healing. The body will heal itself. So this is all controlled by the WBCs. So there are 5 types of WBCs: the basophils, the eosinophils, the neutrophils. These 3 are called as granulocytes. If you look at their cytoplasm is grainy. But these 2, lymphocytes and monocytes, their cytoplasm, on the other hand, is relatively clear. So they are called as agranulocytes. So you can see monocytes and lymphocytes are agranulocytes. The eosinophils, basophils, and neutrophils are granulocytes. So let's study them in detail. The first granulocyte, which we are going to do, is called neutrophil. Now the neutrophil has 62% of abundance. That's a large abundance in terms of all WBCs. 62% of the WBCs are only neutrophils. These neutrophils have a nucleus with 3 to 4 lobes. You can see 3 distinct lobes are very visible. They are formed in bone marrow of bones. They stain with a neutral dye. Because they stain with a neutral dye, the name is neutrophil. It is this which accounts for their name. These neutrophils are basically involved in phagocytosis. So I told you they eat the germs or the bacteria which enter. That is called as phagocytosis. The second granulocyte which we are going to do is called eosinophil. Eosinophil is having 2.3% of abundance in the WBCs. It has a nucleus with 2 lobes. Only 2 distinct lobes are there. Another thing about it is that the cytoplasmic granules are quite large, very distinct and very large. They are involved in phagocytosis, that is eating the germs, antitoxin production. When germs release certain chemicals called as toxins, to counter those, the eosinophils produce antitoxins. And it is also involved in allergic reactions. Allergic reactions are reactions of the body to normal substances, which are not germs. You are allergic to a particular food item. You are allergic to dust. You're allergic to pollen or feather or any food particular item of eating, egg, honey. That is an allergic reaction which is controlled by eosinophils. These eosinophils are also formed in bone marrow, and they stain with eosin, which is an acidic dye. Now, eosin is an acidic dye. Because of staining with eosin, the name eosinophil is there. The third category of granulocyte is called as a basophil. The basophil have only 0.4% of abundance. These basophils have a large nucleus, but the nucleus you cannot distinguish how many lobes are there. The number of lobes are indistinct. It releases histamine for inflammation. Inflammation you already did, swelling, redness, then accumulate the warmth which is generated, and it dilates the blood vessels. When the blood vessels will be dilated because of histamine, more blood can flow to an area which is injured. More RBCs means more oxygen. More WBCs means a faster and quicker fighting with the infections. Then they are formed in bone marrow. Now, they stain with methylene blue. This methylene blue is a basic dye. Because they stain with the basic dye, it's called as basophil. So you have these 3 as the granulocytes. Next, we have the agranulocyte. The first agranulocyte is the lymphocyte. So here, this one matures in the bone marrow. So this is B lymphocyte. This has 30% of abundance. Okay, amongst the WBCs, 30% of WBCs are agranulocytes of the lymphocyte category. This is the smallest WBC. Its nucleus is one, single and large. It produces antibodies to counter the germs or the antigens of the germs. And this is formed in bone marrow in lymph glands like spleen and tonsils. Spleen is a large lymphatic structure present behind stomach. Tonsils are present in the neck region, and they are formed also in the red bone marrow of your long bones. The second agranulocyte is a monocyte. Monocytes have one characteristic, that they are the largest amongst all the WBCs. Amongst the 5 types of WBCs, they are the largest. These have 5.3% of abundance. They have a large nucleus, and that nucleus is kidney shaped. You can see the shape of the nucleus. They are also formed in bone marrow. Now, one property of monocyte is whenever they encounter an infection, the monocyte transforms itself into a macrophage. Once it has transformed into a macrophage, then it will ingest the germs by the process of phagocytosis. So these are the properties of a monocyte. They have the largest WBC with a kidney-shaped large nucleus. They are formed in bone marrow. They transform into macrophages, and they ingest germ at the site of infection. Then we have the two terms which we have been continuously using, the antigen and the antibody. I need to tell you what is an antigen. When any germ enters the body, that germ has to be identified as not a normal constituent of the body cells. So this coating on the germ, that is called as an antigen. It will tell the body, it will tell the immune system of the body that this is not a normal component. So against this antigen, your immune system, the WBCs, start to make antibodies. So the antigen is any substance that causes your immune system to produce antibodies against it. It means that your immune system does not recognize the substance and it is trying to fight it off. So it's a protective mechanism developed by the body to make antibodies. So antibodies are one category of proteins called as immunoglobulins. They are Y-shaped, and these are manufactured by the WBCs to fight against the antigen. And once they have fought it, the bacteria is rendered harmless. So how antibodies, they actually work, they identify the antigen and they attach to the germ cell. Once they have attached to the germ cell, they make the germ cell harmless. Now the germ cell cannot produce the disease. So this is how the antibodies destroy the germ cells and protect your body from infections. Now, this antibody production is used in treatment of certain diseases. We are all vaccinated as children against various diseases. This vaccine is prepared artificially. And how is it prepared? So I'm using the animal here as a rabbit. These days, we use the bacteria called E. coli, Escherichia coli, but this is just a sample experiment to make it easy for you to understand. So first of all, the antigen, the disease organism, is injected into a healthy animal like rabbit. Once the animal will be injected, the immune system will obviously trigger a response against that antigen and will start producing the antibodies. Once the antibodies are produced by the lymphocytes, they accumulate in large number, the animal recovers. Now, once that animal has recovered, the blood of that animal will be rich in the antibodies. So we extract that blood and we allow that blood to clot, producing a serum. This serum will be rich in antibodies. So this will be called as anti-serum. It is this blood serum containing antibodies which is used against a specific antigen. So when we are vaccinated against particular diseases, this is one method by which the antibodies prepared from another animal are used in your body. This is one type of vaccine which is prepared. The second way in which the WBCs can help is in preparing antitoxins. Now, just like you have antibodies being prepared, same way antitoxins are also produced. This was first of all observed and discovered and made use of in the year 1840, when they found that diphtheria, which was a very deadly disease at that time, this could be cured by simply injecting horses with diphtheria toxins. So what was done? The diphtheria causing bacteria was grown in the lab and its toxin was harvested. So it was collected, purified, and this harvested toxin was then injected into a healthy horse. So this was injected into a horse. After it was injected, the horse would obviously fall sick. The immune system in the horse helped it to produce antitoxins. And the horse, after falling sick, would recover. Once the horse would recover, then the scientist collected some blood from the horse and separated the antitoxin rich serum. So this blood was allowed to clot in a test tube or a container, and the clear serum above was collected. This serum was rich in antitoxins. So researchers purified the toxin antiserum, and this could then be used in the treatment of the disease called diphtheria. So this was used for diphtheria at that time when the study of immunity, the study of immune system and immune response was just developing. So here, these days, we do not use animals, we prefer to use bacteria, but they're very simple examples to make you understand how antibodies and antitoxins are synthesized. So with this, we have reached the end of the WBCs. So you have done the 3 types of granulocytes, the neutrophils, the eosinophils, and the basophils, and the 2 types of agranulocytes, the monocytes and the lymphocytes, and how these bring about the immune reactions and the immunity, control the immunity in the body. So I hope you have understood it all. In the next video, we will be covering the platelets. So here, I hope the concept of white blood cells is pretty clear to you. So happy learning to all of you. Thank you very much, and God bless you all.